Hi class, this is a video on the cell membrane. In our previous video on the lipid notes, in lipid notes we talked about parts of a cell membrane. So cell membrane is the membrane that's found outside of a cell. It actually has a very similar structure to the nuclear membrane as well as the membranes around like the mitochondrion, the endoplasmic reticulum, the Golgi. They all have a very similar membrane around them. And these membranes all have lipid molecules. So in the lipid notes we talked about phospholipid molecules. So we have a bunch of phospholipid molecules that are sitting next to each other in a lipid bilayer like this. So this is what we talked about in our last video on lipid notes. Now each of these phospholipids has a head and it has a tail. And if we look at the head and the tail a little bit closer up, we know that the head has a charge on it. So a head has a charge, has a slight negative, slight positive in a different area. So the head is hydrophilic, which means that because it is charged, water is going to be attracted to it because water is also charged. The tails are hydrophobic, which means they don't want to be by water, which means if we dropped it next to a pool of water, here's my water here, we drop these structures that they're going to automatically turn and line up like this so that the heads are near the water and the tails are away from the water. So if we're looking at a cell, we have water on the outside, we have water on the inside, so that's what forms this lipid bilayer. So we talked about that in our last video. The majority of the cell membrane is made up of a lipid bilayer. Now there are other molecules we're going to talk about in this video that are also found in the cell membrane, in particular proteins. So before we get into that, just the function of the cell membrane. So the function of the cell membrane can be protection. So it kind of like acts as a barrier that keeps stuff in as well as doesn't let stuff outside get inside of it. It serves as a site of communication with other cells. And that's what we're going to talk about with the proteins today. So the proteins help communicate. So when cells bump into each other or release chemicals that bind to another cell, that's how they communicate with each other. Now, you can talk about cells communicating in a multicellular organism like us, like our cells, like our pituitary gland sending out hormones to communicate to other cells, telling them to grow, for example. Or you can talk about single-celled organisms like bacteria. So bacteria releasing chemicals so they can communicate with the cells that are near them. The cell membrane also selectively allows substances in and out of the cell. So it's selective about what it lets cross. So for example, if something is too large, it might not be able to cross at all. So things that are small might be able to cross, but it depends whether or not they have a charge. So small and neutral can easily cross over. Small and charged needs a little bit of help. Small and charged negative needs a little bit of help. So it's selective about what it lets go in and out of the cell. It responds to the environment. So that's particularly true in the case of like single-celled organisms like bacteria. So it can detect environmental cues or chemicals in the environment and react to it that way. But that can also help inside of our bodies as well, like in particular like white blood cells. So white blood cells may bump into a foreign invader and then react to it in its environment. And then the last thing is the cell membrane functions to recognize it as self. So self. So it knows that self based on like the protein shapes. And I'm just going to simplify it here and draw triangles. So that's how I can recognize it as itself as self is by the shape of the proteins that are found in the cell membrane. So quick review on phospholipids like we talked about in our last notes on the lipid notes. We know that they have hydrophilic heads, so this is the head here, and it's hydrophilic because it has some charges on it. And water, remember water has charges too, so water has a slight negative end and a slight positive end. So because of that, they're going to be attracted to each other like magnets. So the heads will always turn towards water. As they turn towards water, they'll be pulling the tails away from water. So the tails are considered to be hydrophobic. Again, remember the tails themselves are not repelled by water. It's just because the heads want to be by the water so much, it'll turn, so the molecule will turn towards the water and the tails will then turn away just because the head is turning towards. And what phospholipids do, they make the majority of the cell membrane. And they do control what goes in and out of the cell. So we have our lipid bilayer. And like we said, these molecules are not physically attached to each other. They're just sitting there, and they're sitting that way because of the charges. So because of that, you can have things that can cross over the membrane directly. Now, in order to cross in between the phospholipids and kind of squeeze between there, it has to be small. So large things cannot cross this way. Large things can either not cross at all, or they have to cross over like a bridge, like a protein bridge, to do that. So things that are small can cross this way. Things that are small and charged, for the most part, cannot cross this way, because something with a charge is going to have a hard time getting past the heads. It's going to want to stick to the heads. It's not going to want to continue to move through. But some things like water can actually go through. It's small and it's charged, but it goes very slowly because it really wants to hang out with the heads. It doesn't want to go through the middle part of the membrane there. So phospholipids make up the majority of the membrane and control what goes in and out of it, just strictly based on having those charges. Now, because these molecules are not attached to each other, so here we see a phospholipid, here's other phospholipids, the other side of the lipid bilayer here, and even the protein molecules that we haven't talked about yet. Here's a bridge right here. These protein molecules here, none of them are physically attached to each other. They strictly sit the way they sit because of charges. So we know that the heads are charged. I'm just going to put negative here. So that means this part of the protein is also negative. So heads down here are charged. This part of the protein is also negative. And that means in the middle, it's all uncharged. So hydrophobic right there in the middle. So because they are not charged, not connected physically to each other, then things can cross, they can like squeeze through here. And that makes it such a great design. Because they're not connected to each other, they call it the fluid mosaic model. So fluid mosaic model is just referring to like the motion that you see cell membranes go through. Talking about proteins in the cell membrane, so there are proteins that go all the way through the cell membrane, like this one right here. So this one would be charged on the ends and neutral in the middle. There's some that just sit on the outside here, which means that they're going to be charged, whether it's positive or negative, doesn't matter. So this one also sits on the outside, so it's going to be charged as well in those areas. We're not going to talk necessarily about how they have different functions, just functions in general. So let's take a look at some functions of these proteins that are found there. So some of the proteins serve for cell recognition. So here's our lipid bilayer right here. So I'm just circling a couple phospholipid molecules. And here is our protein molecule here in blue. You can see that it also has these little small hexagons here. These are sugar molecules. So the protein has these sugar molecules, or a carbohydrate chain, because sugar is a carbohydrate, attached to it. And this is how, when other cells bump into this cell, that's how they recognize it as self or non-self. They're like, oh, that's the right shape, and that's self. Oh, it's not the right shape, there's non-self. Now we have thousands of different cell shapes that are acceptable. And our body takes a survey about what our self chains look like before you're ever born. So you have a lot of them, and it's based on just these carbohydrate chains is to identify as self or non-self. Now, if it's non-self, then you're going to have an immune response where your immune system's going to attack that cell. So that's one thing. And these marker proteins here identify it as self or non-self. So here's a picture of a couple of different shapes of marker proteins. So you can see here that the carbohydrate chain looked different than this carbohydrate chain did right here. And this chain looks different here. So you're going to have a lot of different variations of it. But again, your body takes a survey of all of your variations before you're ever even born. Another function of a protein that's found in a cell membrane is something called a receptor protein. So in this case right here, you can see here's the receptor protein here in blue. And you can see that it has a binding site here. And the binding site fits only a certain chemical, like a lock and a key. So not just anything can bind there, only this specific chemical. And the chemical that comes in and binds to receptor proteins are called ligands. Common ligands are hormones. So as you're producing growth hormone, for example, it's going to go and it's going to fit in only in the cells that have this specific receptor that fits it, and then it's going to stimulate those cells to go through cell division. So a ligand, typically hormones that influence other cells in your body to have a response. And that's in a multicellular organism like ourselves. But bacteria can do this as well too. So bacteria can release chemicals that influence cells that are nearby as well to cause some sort of a reaction or response in that cell. The third kind of protein is something called a channel protein. So I mentioned it's kind of like a bridge. So you can see that it's designed where things can cross over it. Now, the things that cross over channel proteins can be large. They can't be too large because you can see there's a size limitation. But they can be larger. So maybe they can't fit through here because maybe they're too big. So maybe that can't happen. So they go through a channel protein. They can also be small but charged. So if they're like an ion, for example, so say it's a sodium ion. So a big sodium ion is small, but it can't pass by these heads. These heads will not let it pass because that's a full charge. So it won't let it go through there. So instead it has to go through a channel protein. So if a cell didn't have a channel protein, it can't transport an ion. It can't transport large molecules. So that's why cells have channel proteins to help enable move certain things in and out of the cell. Okay, and that's the end of the notes, and I'll talk to you soon.